uh, invited talk of this fourth day of the Wabio. For me, it's a very, very big pleasure, and I'm very excited to introduce Peter Stadler. Professor Peter Stadler leads the group of bioinformatics at the University of Leipzig, but he actually is affiliated to the Max Planck Institute of Mathematics and at the Fraunhofer Institute for Cell Therapy and Immunology. As you can see there, he's also associated at Institute for Theoretical Chemistry at the University of Vienna and the Center for Non-Coding RNA in Technology in Copenhagen and the Santa Fe Institute. He's an external faculty there, and he has been given the honoris professor honoris at the National University of Colombia as well. Um, it will take me like the whole hour to talk about Peter because he has a very, very long CV, but just a little bit. His undergrad background is in chemistry, but he also studied physics, mathematics, and astronomy. And he also has this master's and PhD in all these fields. In all these, and he has tried not to get more of these degrees, but he continuously is learning and getting into new projects. He's one of the pioneers of the study of the RNA secondary structure and of many of the softwares that we are using nowadays. Well, he's like, let's say, the leader behind it. So I would prefer that Peter tell us about these findings and his research. And thank you very much, Peter. Peter is coming all the way from from Germany to Guatemala to attend our group retreat that we had last week over there, and now he's here just because of the Wabio. So I appreciate very much, Peter, that you are here today with us.
Thank you very much. Do I have to do anything to switch this on, or does this work? Okay. Yeah, you never hear this yourself very, very well. Maribel, thanks so much for the kind introduction. It's always a pleasure for me to come to Mexico. So the first picture is this is the main building of Leipzig University. So this is just to show that Sinve stuff here is not the only place with a cool building. But what I want to do is I want to talk about RNA and in particular RNA secondary structure prediction. And I have to apologize a little bit to the people attending online because this talk is a little bit integrated with what we will be doing in the course afterwards with Katia Avila. So I kind of skipped the biological introduction because Katia will be speaking about this. So our object of desire is ribonucleic acid RNAs. RNAs are very peculiar molecules in the sense that they, similar to DNA, are made up of just a small alphabet of 4 different nucleotides, but very different to DNA. They do not appear as very boring double strands that just make one very, very, very long helix and don't do much to a first approximation otherwise. But instead, RNAs are usually single-stranded, and that means they fold back onto themselves. And they do this in a very characteristic way that allows us to do a lot of detailed mathematical straightforward modeling. And the rules for RNA folding are actually very simple. There is only 3 types of base pairs that can be formed. You can see the base pairs here in this diagram as this kind of parallel arrangements of two of the nucleobases from the different sides of the molecule. And each nucleotide either pairs with something else or it doesn't. So in that sense, we can think of the pairings just as a matching in a graph. In addition, if you think about this very long string of RNA, if this folds back onto itself, and as you can see, whenever it folds, it doesn't make a nice parallel ladder, but it actually, like DNA, forms helical structures. So once you form these helices, right, that drills up, winds up the structure. So once you have a helix that kind of prevents you from threading anything else through these loops. So what that does is it allows us to approximate RNA structures by something where we have an outline on the outside and base pairs only on the inside. So base pairs, in a sense, cannot cross. Where they can, this is called pseudonodes, but we will ignore this for this presentation. And this can also be expressed as a very simple condition. I'm not going in this presentation into any of the math of RNA folding. I will torture the students excessively for the rest of the day with this. So this will be our main topic on how all the engines behind the scenes that I'm talking about here actually work in detail. What I want to do in this presentation is to talk about a few extensions, about a few new things that we built into the RNA secondary structure prediction machinery over the last 3-4 years, basically. I will need a little bit of notation because this will appear in many of the slides. This no crossing condition essentially allows us to write RNA secondary structures in a very nice linear representation where every base pair is formed by matching parentheses. So parentheses that match up like in a mathematical expression, right? So this opening bracket corresponds to this closing bracket, or here, this opening bracket corresponds to that closing bracket. So the point is that you can just go through this structure and find for every opening bracket what is the corresponding closing bracket. So in that sense, and therefore identify the base pair. Since RNA secondary structures are just defined by their base pairs, that means these dot parentheses strings are just faithful, a one-to-one proper encoding of the secondary structure. So we can use this as a way of representing structures instead of drawing them in this more conventional way like this here. What we would like to do, of course, as structural biologists, would be to calculate or to compute, predict the actual three-dimensional structure. Now, this is a much harder problem than secondary structure prediction, it turns out, basically because of the fact that secondary structure prediction is relatively easy. Secondary structures capture most of the energy of the structure formation. They are usually axial folding intermediates. So what you get is you get a structure that essentially has the helices, which is these colored parts, and the loops exposed, and only afterwards at a longer time scale the structure actually folds up in the three-dimensional structure. And the additional energy that you get is actually comparably small. So in that sense, RNA prediction, RNA structure prediction is very different from the problem of protein structure prediction. Now, since there was the Nobel Prize for AlphaFold, I have to stop here and add a brief footnote on why are we still doing this kind of stuff with physical modeling for RNAs and not build an AlphaFold for RNAs and be done with it. There is actually two reasons for that. The first reason is in RNA, we are very often also interested in predicting structures, in designing structures that are not natural. And what AlphaFold does, it very well predicts structures for sequences, for protein sequences that have evolved for a very long time, and it does so knowing the rules of variation in natural sequences. It actually doesn't solve the folding problem, which is sometimes the kind of public announcement, right? It solves the folding problem for evolved biologically relevant structures, and it does that very well. I don't want to denigrate this at all, but it does not solve the physical problem of predicting the structure that comes out if I give you an arbitrary amino acid sequence. So this is the one reason that also we have here. We want a more physics-based model because we are also explicitly interested in the RNA field, in structures of RNA molecules that have not evolved, that come out explicitly from rational design and have nothing to do with natural sequences to start with. But the real reason why we don't do this is we simply don't have the data. For proteins, you have a very large number of very well determined protein structures from X-ray, from NMR, recently also from cryo-EM techniques. For RNAs, we don't have these, and the reason is mostly technological. RNAs don't particularly like to crystallize, so getting crystal structures for RNAs is let's say, a very complex procedure. For a while, it was fashionable to find RNAs that crystallize well and get their structures, but of course, they are very, very special structures that you don't generalize very well. And the other technique that's usually used, which is NMR structure determination, also does not work well for nucleic acids, and the reason is because we, instead of 20 amino acids that we can distinguish, we only have 4 different letters, so it's very difficult to assign which signal comes from which nucleotide. And along the backbone in nucleic acid is this sugar phosphate chain, and essentially there is no coupling across the phosphate group, so you also cannot get adjacency information between neighboring nucleotides. So that means the only way to get NMR structures is to do isotope labeling with stable isotopes, and actually lots of different isotope labeling that makes it expensive, tedious, and you just don't do it if you don't absolutely have to. The reason why I'm talking about is that towards the end of my presentation, we will actually be talking about using external experimental evidence to guide secondary structure prediction. And the reason why and this is still a very active, timely topic, exactly because we only have very, very few real three-dimensional structures. So almost all the experimental techniques that go into determining RNA structures are actually chemical methods combined with sequencing. We get to that give you constraints, that give you hints, but that don't give you a structure directly. So that's the part of the story that I want to focus on today. So we had these. So how does RNA folding in general work? The idea is that we actually will minimize energy. And we will do this on a discrete model that only looks at base pairs, but in reality, the energy of RNA or the stability of RNAs is not determined by the individual base pairs, but actually by the stacking of 2 base pairs onto each other. And this can be understood basically from this 3-dimensional picture here again. So if you have a base pair like this, what you have is you have a bunch of hydrogen bonds between these nucleobases. And in an aqueous solution, whether you do the hydrogen bonds between nucleobases or to the water, energetically makes very little difference. So the base pairing itself is not the point. the point is that you have these big aromatic side chains that in parallel stack on top of each other. And this stacking interaction that comes from, for the chemists among you, pi electron clouds of these large aromatic molecules, so that sit basically between these whole systems here. This is what stabilizes the structure. And then you get, I'm not patient enough for this. So, and then we get destabilizing effects from all the loops. This is basically loop strain. And what people have done is they have produced small molecules that essentially can only form one particular structure, like a hairpin shown like this with particular base pairs, and then meticulously varied the individual bases here and measured melting curves from them. So actual physical-chemical melting experiments that give you directly energies and entropies for folding and unfolding these structures. This is what our data come from. So we have data essentially for the contributions of these little pieces, and then we make the assumption that the energy of our RNA is just the sum of the energy of all these little local loop-like contributions. And this is not my hieroglyphs. This is a representation that we will look at in the course. This is basically a way of writing down how these RNAs get decomposed, but I promised we will not be talking about this here. So I will gloss over these and go into the things that we can do with these RNA structure predictions. So one of the things that have become very interesting in the last years is that people realize that RNAs not only interact with themselves and fold back and form their structures, but they also can interact in the same way with other RNA structures. And the nice thing now is, since we have a physical model that says how do pieces of RNA pair, we can actually use the same model to ask how do RNAs interact with each other. Well, they interact with each other by just forming base pairs between strands that come from two different molecules. But for the base pairs that are formed here, right? So you have a stacking of base pairs that just come from the red and the green strand. As far as the energy of these stacking interactions is concerned, this is the very same thing as if you would have these interactions within the same molecule. So now we can ask, can we actually calculate RNA structures that come from bringing together two or more of these different RNA strands? By the way, one of the key examples of a multiple interaction of RNAs is the spliceosome. In the spliceosome, you have several different RNAs, spliceosomal RNAs, that during the splicing reaction actually interact with each other in different ways and form sometimes complexes of 3 of them at the same time. And one can actually calculate these interactions with exactly these machineries. You just calculate secondary structures for multiple strains. So there is a little technical subtlety, again, something that I will gloss over. Just, I don't know why this thing always thinks it has to jump. So this is, so if we just have 2 strands, then there is nothing complicated. If we have 3 strands and we want to calculate the structures from them, sort of using secondary structure calculations, as we have them, then the problem is it starts to depend on the order. But there is a very nice theorem that says if we have connected structures between different pieces of RNA, then we can distinguish on whether the base pairs that are formed are the same or are disjoint sets of base pairs. And that depends only on whether the two permutations, so I give you two different arrangements, two different ways of ordering my, say, 5 different RNAs, and all I have to check whether the combination of these two permutations is cyclic or not. And then either I get the same structure and I don't have to calculate the base pairs, or I get a disjoint set of base pairs from all the other structures, so I have to calculate exactly all these base pairs and then minimize over them. So this simple result allows us to calculate, forget the stuff on the bottom, we will come to things like that in the course. So there is a simple way of arranging individual RNA bits and pieces and just use the same machinery that we have for RNA folding. And this theorem basically tells us over which combinations we have to summarize. Come on, back 1, back 2. So these RNA interactions we can calculate, and one can actually rewrite these recursions again. We will see how that works later in a very fast way. This is annoying. Go back. So this is basically to show that it works. We can do the computation much faster than everybody else. So this is kind of the usual stuff that you're supposed to be showing as a bioinformatician once in a while. If you advertise tools, right, your tool does the right thing and it does it much faster than everybody else's. I don't want to belabor these here because I always find these. What the beep? Okay. One problem that has plagued RNA folding, so RNA secondary structure predictions, for a long time was that the parameters that have been measured were actually measured under conditions that are not very physiological. So usually it was 1 normal sodium chloride, so very high salt concentrations, which sometimes is a problem in detail with the accuracy of the predictions. So why is this important? So why is salt concentrations important? The backbone of RNA, as I said, is this sugar phosphate chain, and this phosphate is negatively charged if you put the RNA in water, so you get essentially a very long chain that's an anion that's very highly negatively charged. So of course you need counter ions in your solution, and this is usually supplied by salt in the environment. And what happens then is that so because you have this negative charge that's in particular also not homogeneous over the whole structure, but concentrated in the grooves, what you get is that the positively charged ions, the salt, in this case sodium, arranges itself also in a very specific way along the backbone. And what that does, if you look at models of electrochemical models of these polyon ions, is that this behaves like the cations that are kind of glued to the negatively charged backbone masks some of the, but not all of the charge. So you have to basically deal with effective charges that are left in this molecule, and these effective charges lead to a an electrostatic repulsion effect that let's see if this works better with my fingers. So leads to an electrostatic repulsion effect that becomes larger and larger the larger the charge is, so the smaller the concentration of the counter ions is. And this is something that not my lab, but a group in Berlin some 15 years ago actually was looking at in a lot of detail. And they derived this absolutely horrendous, ugly formula. But the point is, this is just a function that you can teach the computer to evaluate. This doesn't hurt particularly. This K0 is also a Bessel function that you can evaluate. And this whole thing is stuffed with natural constants and geometric constants of RNA. So the length of the helical rise, which is about 3.4 angstrom, goes in there, and then some good old friends from electrostatic theory in solution, like the Bjerrum lengths, the Debye screening lengths, and whatever goes in there. And the only thing that's really important is that there is only one particular point where the salt concentration actually enters. It enters into this factor kappa, which is also sitting in the Y. So at the end of the day, all you have to do is evaluate these disgusting functions as a function of the salt concentration. And to bring these together with the, this works much better, and to bring this together with the measured energy parameter, all you do is that you basically say, okay, so I calculate the ugly expression for the salt concentration under standard concentrations and under the concentrations that I have. And all I do is my salt concentration relative to the measured energy model is simply the difference of the salt concentration that for my conditions that I'm interested in minus the salt effect for the standard conditions. So that gives you this correction factor that one can actually calculate. And the interesting thing is in this correction factor there is no empirical measurements or anything in there except for very simple geometric parameters like the helix size of the RNA itself. So except for a little bit of geometry of the RNA molecule, nothing goes in there. There is one fudge factor that we have to do is that if we have two different molecules that come together, they also displace the ion cloud, and that is also concentration dependent, so we need an extra term for this. The salt concentration effects can be relatively large if you go to large loops and small salt concentrations. So the smaller the salt concentration, the larger the destabilizing effect, both on the loops and on the stacking. So the unit is slightly old-fashioned kilocalories, but these kilocalories per mole. For whatever reason, the RNA community never managed to adopt proper SE units, so they just are stuck in their kilocalories per mole, so we just also use them. For comparison, KT, so thermal energy, is about 0.6 kilocalories per mole. So this is about 6 times thermal energy or something like that. So that's already a sizable effect on the structure. So what we see is basically if you make salt concentrations just a bit smaller than the standard concentrations, say you go from 1 molar to 0.1 molar, the effects are rather small. But if you go to very salt poor solutions, which sometimes you do in the lab, then the secondary structures will actually be affected because these energy contributions become reasonably large. There is also some experimental data where people actually looked at folding energies, binding energies in RNA as a function of salt concentrations. And you can see that the experimental data and the prediction from this model as we've built it into the RNA package correlates very nicely. In particular, it works very well also for these larger hairpin-like structures. So the energy differences that we get here are pretty much what we expect within the experimental error. So you have a few tenths of a kilocalorie measurement errors usually in these experiments anyway. This is basically just one tRNA structure where we, for the fun of it, just artificially showed how the structure would change. So this is basically under standard conditions. This is under more physiological conditions, which is pretty much still the identical structure. But if you reduce salt concentrations more and more, the structure starts to fall apart because the electrostatic repulsion starts to take over and remove the structure. Another topic that I want to go into very briefly, so far I talked about RNA's secondary structures just being base pairs. But there is some other structures in there that kind of behave like base pairs, even though they're a little bit more complicated, that we still also can take into account. And this is so-called G quadruplexes. So G quadruplexes are formed essentially by runs of guanosines. So if you have a very G-rich local piece of sequence, then what that likes to do is produce these layers of guanosines that are stuck on top of each other. On the next slide, I have a nicer picture how this looks like. So you have these guanosines that actually don't form Watson-Crick, but rather Hookstein-Watson Crick bond combinations. They are centered around a metal ion in the middle, usually a potassium. And what they do is they kind of stack on top of each other. Again, it's the stacking energies of these pi electron clouds that holds the whole thing together. Quadruplexes have been implicated in a lot of functions, so they also exist in DNA, where they appear in telomeres, in promoters. They seem also to interfere with protein functions. There are proteins that specifically bind these quadruplex structures. And for RNA, there is an ever-increasing list of functions that have been reported for these quadruplex structures. And they also are important for dimerization in viruses, for instance, in HIV. They control lipids in transcription in bacteria, and they are also in mRNAs. They are ligands for several different proteins that specifically recognize quadruplexes and use these as part of the gene regulation machinery. In the RNA package, we have also included the possibility to compute quadruplexes. This works basically because a quadruplex is a local structure that basically technically looks like an RNA hairpin, and so it can be easily included into these structures. We didn't really bother to make a very nice graphic for that, so it basically appears as a loop with a bunch of extra interactions that it identify the layers in this dot plot representation. Again, something for the course later. They appear as triangles, so one can calculate these quite efficiently. These I want to gloss over. So we can also do these now in circular RNAs. You've probably heard that more and more circular RNAs have been found and implicated in various different functions. They appear essentially as byproducts or as deliberate products out of the splicing process. And so now one can also look for these quadruplexes in the context of circular RNAs. This was not possible before, so people haven't looked at that systematically. What I should also emphasize is that so far I've spoken as if there is one sequence that makes one structure and that's the end of it. Right? This is a little bit the picture that you have in mind as a biologist. There is my RNA, ribosomal RNA, tRNA, or whatever, has this structure. Nature doesn't know that. What actually happens is that every one of the structures, be that a protein or an RNA, actually forms a whole ensemble of related structures with very similar energy. And here is just one essentially designed example to show the effect. We have here the sequence, we have here the ground state structure, so the most stable structure, and then I just wrote out all the other structures that this molecule can form within 1 kilocalorie, so within 1.5 times thermal energy range. So these are the structures that for sure for this molecule would be present at least a few percent fraction in a test tube where you throw this sequencing. And as you can see, there is one part of the structure that is very well defined, where they all agree that it's a good idea to make this hairpin. So all low energy structures make this hairpin, all the low energy structures make this loop consisting of, or this helix consisting of 5 consecutive base pairs that clamps the ends of this molecule together. But then there is another very variable part of the structure where essentially anything goes. So this is the typical picture for these RNA structures. And the nice thing is that this is also something that we can calculate with this RNA folding software directly. You can have all these things enumerated, or you can have summarized these into the probabilities for the individual base pairs. And then you get plots like these. Down here I've drawn, so the way how you read this is we have the sequence here twice, and every dot is a base pair between the second position and the last position, or the second to last position and the first position. And down below I only showed the most stable structure, which has a loop at the end, and then essentially 3 different loops, 3 different helical structures. So this looks like vaguely like a tRNA, the horsing. And then I can also, for every base pair, calculate what is the probability that this base pair is actually present. And this is what I'm drawing in these so-called dot plots. And you can see here there is sort of the big fat diagonals are helices that are present in almost all structures, and then you have a lot of helices that can be formed as alternatives. So this is how you read these structures. And this idea that we have multiple structures that each comes with its own energy is the important starting point for the second part of my presentation, which is basically the question, how do we get experimental evidence into these secondary structure predictions? So far, we've only used the thermodynamic model and not in any way looked at any other evidence that we could measure. So we know, of course, this thermodynamic model is an approximation. It cannot capture everything, and it cannot capture every effect that we see in these RNA structures. So what can we do with additional information? First of all, let me very briefly, so relax, not much math. This is actually a few biology slides. Now, there is various different methods that try to assess RNA structure experimentally. One of them is shape analysis. So it starts with the usual, we prepare everything in the lab until we actually have our E. coli. And then what we do is we expose the RNA to a reagent like this thing, NMIA, but there is also dozens of different alternatives that one can use. And what basically happens is that if you have a flexible piece of RNA, so where basically this OH bond is easily accessible, then this reagent can actually bind very nicely to this position. You get an RNA structure that has in certain positions, namely those that were particularly flexible, you get this large organic residue as an adduct. So it's literally sitting at the nucleotide. And what that does then is, if you try to sequence this RNA, this will actually lead to the reverse transcriptase to stop. So you basically measure positions of stops in your RNA. And essentially what one measures is the more stops, the more read ends you get at a particular position, the more signal you get. And for shape, a certain reactivity score that one can calculate has been proposed. And if you look at this piece of RNA that was sequenced for this particular experiment, then you will see, okay, so regions with very high reactivity exactly correspond to the loops, and other positions with moderate to high reactivity are also associated in the loop. So this is the type of information that you can get. I get the signal that basically tells me which positions are flexible, and flexibility basically means this thing is essentially unpaired, or it's likely to be unpaired. And then I have for some positions in my RNA, I know, okay, so they should now appear as unpaired positions in my structure predictions. So what I want to do is to add this then in my structure prediction. There's a bunch of other protocols. A particularly nice one is this parse protocol, because it uses a digestion by two different enzymes, one that likes to cut double strands, the other one that likes to cut single strands, and then you can basically calculate the log odds ratio, which is part four, which will tell you whether the position is more likely paired or more likely unpaired. So you get evidence for both paired and unpaired positions. Again, an example of what this data look like in practice. So you get for some positions shown in red here with the red arrows, these are likely unpaired regions, and you have the paired regions, and you have the greenish things which are likely paired. But what you also can see is, if this is the only evidence, if this is everything that you see for your structure, right? This does not give you a complete structure prediction. This gives you constraints. This gives you a hint. This nudges the structure. This could potentially be used to nudge the structure into the right thing, but it's not a determination of a structure. With the biochemists in Leipzig, we also developed our own protocol for this type of chemical problem. It's called LEDSEC. It uses lead cleavage. And the trick of that thing is essentially that in order to get much cleaner data than other protocols, we actually sequence the fragments from both sides of the cleavage, which allows us basically to get rid of false positives that come from other biochemical processes. So this thing actually works reasonably well also directly in vitro. I'm not going to go through the whole process of the biochemistry. So basically what you do is you take your RNA in vivo or in vitro, you expose it to lead, and then you process it into two libraries, one that looks at the phosphate end, one that looks at the 5-OH end of the cut. And that gives you then reasonably nice signals. So this is for a tRNA as an example. What I want to point out here, whenever you have a protocol that works but with sequencing as the readout, say you cut and cutting the sequence. So you have an experimental protocol where your readout is chemical modification or direct cleavage or whatever it is that cuts the molecule into parts. You have the problem that you don't see one end of the molecule because the fragment that you cleave off is so short that you can, in the sequencing, you will see it, but you cannot map it back to a genome, right? You cannot identify where this came from if you just have 5 nucleotides. This is too short to identify what it was, so you cannot analyze it. So essentially, so this is why this for the phosphate side, you don't see the 5' side of the molecule. You only can start mapping things. This is for E. coli. When the fragments are about 14, 15 nucleotides long, everything shorter does not unambiguously map to the genome, so you cannot say where it comes from, so you cannot use it. And the same thing if you use the 5-OH library, you basically lose the 3' end of the molecule. So the nice side effect of using both libraries in our protocol is that you actually get the complete coverage with at least one library, so you get a full structure determination excuse me, of the molecule from both sides. So combined, these things actually give you the full signal. So how do we analyze such data? We have a thermodynamic model that is based on energies, right? So we have energies for these loops, if you remember what I started out with. So what we have to do is we have to convert the signal that we get from the experiment into something that we can put into this energy calculation. And the rational way of doing this is to say, well, we have a pseudo energy that essentially comes from a log odds ratio that basically says my reactivity, what is my probability that a particular position is, let's say, unpaired, divided by that it's paired. And in order to get this in the same scale as a thermodynamic energy, I just have to multiply the log odds score by minus RT. So that converts the observed data into something that is an energy at the same scale as the energy parameters that we have. So the question then is, we have to take our original data, our original signal, say shape reactivities, and we have to first convert these into something that fits the secondary structure model, which is a probability to be unpaired. And that we can then convert into an energy contribution that we can directly plug into the structure prediction. So for shape, this was usually done with some regression model, which is a little bit of voodoo. but one can make this more rational, and so we actually tried, and the protocol that I will show you in a moment actually works at least as well as the formula that has been used by Kevin Week's lab. But this is a technical detail. So the problem with these signals is that, as we've seen, I think in these examples here, so the evidence is often very small. For many positions, you don't know anything, you don't get any answer, and it's rather noisy on other in other points, right? If you look at these, whether this black thing here is really different from that yellowish peak here is really in the eye of the beholder, right? So this is basically a threshold that you more or less can make up as you want to say, okay, so this is probably unpaired and this is probably not unpaired. So to do this in a more rational way, what you can do is you proceed in two steps. So first of all, we don't just take the read counts wherever they are, but first of all, we normalize the read counts by what is actually the expression in a particular locus, right? So if I have lots of RNA, I will also get lots of cleavage points. So I don't want the absolute number of cleaved RNAs of read ends. What I want is that normalized by expression, and that can be done either internally in the data or you have extra expression data for that. So then we get a normalized signal that says what is kind of the fraction of RNAs that was cut, say in LETSEC, by at position K. So now we can use this normalized signal and ask, well, does high signal always indicate that the position is unpaired, and does low signal always indicate that the position is paired? So what we do is we take RNAs where we know the secondary structure, ideally from a 3D prediction, and just plot how many unpaired positions have a certain signal for being cleaved. And then you look at this and you're very frustrated. So what you basically see, okay, so high signal is more prevalent for unpaired positions than for paired positions, in both types of libraries. But it's not like you get nicely separated peaks or something like that. So we only get a difference in probabilities, but we don't get a nice separation where the signal would allow us to tell this position for sure is paired, this position for sure is unpaired. So that means we cannot just put hard constraints in our structure and say if there is a high signal, then it has to be paired. There is a bunch of cases where this is actually unpaired positions. And vice versa, if there is a low signal, we basically cannot say anything. So this is also one of the important issues here, that you really have to think about what is your signal measuring. In this case, it's highly flexible positions that are likely unpaired. So you essentially, for the paired part, you simply have no signal, so you should not touch this, right? You should not say, if I see a low signal, that has to be paired. This curve shows you very nicely, you don't just know anything in this realm of the curve. So what we can do now is, since we have a few reference structures, we can actually ask, well, given a signal, what is the probability that our position is in fact unpaired? And so you just average that over bins of data that you have, and then you feed a nice sigmoidal curve into it. You can actually also try to capture all sorts of experimental effects on that. So we looked at this in our E. coli data, and what we found there is that for some reason, whenever you have a cleavage site between a C and an A, this is much, much less informative about the structure. The reason probably is that there is nucleases in E. coli that like to cut in these positions anyway, so you just get a lot of background. while for all other dinucleotides that basically looks reasonably nice. So you really want in this particular data set, and this is all things that you can take out of a single data set. So this is all things that you can determine internally. You don't need external controls or anything for these. You can actually figure out, okay, so CAs are something different. I don't want to use them in the same way as all the other dinucleotides. You can actually also use the two libraries together and do a two-dimensional regression on these that improves structures even more. So what do we get out of these? So what we did is we took now this energy model for our LEDSEC data, so for our signal that we get from the LEDSEC data, and ran that over all the RNAs in E. coli for which structures were actually known. And we just looked at Matthew's correlation coefficient as one example of a performance measure. So for everything on this side of the diagonal, there is an improvement from the experimental data improve the prediction of the structure relative to just using RNA folds, so relative to only using the thermodynamic model. So this looks pretty good. There is a lot of things where you really correct fairly screwed up structures that have very low probability. So there is, for instance, domain 4 of the ribosomal RNA, which is very badly predicted. Sometimes it essentially doesn't do anything, which is fine. So on average, we get like 20-ish% improvement in performance accuracy. But there is a bunch of, in this case, 3 conspicuous outliers. And in this case, we actually went after these cases and we found where 6S RNA actually, depending on the state of E. coli, whether you're in the exponential phase or not, has 2 different structures, and the reference structure is actually the that is for the exponential structure, but this was done in saturation. So we are actually right that we get the. So this is basically the wrong reference structure. This is not the structure that we should see. So this is clear that this looks like we get a worse structure because we actually predict something that's much closer to what the structure actually should be. For spot 42, there is an error in the reference structure that one can easily identify. And for the tRNA, this is basically a consequence of a chemical modification. So the outliers we can explain. So in basically what that says, the machinery works, but still even with the effort for doing the experiments, you get something that's still in general reasonably far away from perfect predictions. One reason why that is the fact that if you just determine whether a position is paired or is unpaired in the RNA, this is not sufficient to determine the RNA secondary structure. And I just give you a very simple example here. So this is a made-up sequence to show the effect very nicely. This sequence can fold into 2 alternative structures that look like this. So the upper structure is this guy here that has essentially 3 independent, 3 hairpins. And the lower structure is this guy here, which is more or less a rod-like thing with some extension here. And what you see is both of these structures have the same pattern of paired versus unpaired structures. So it's clear that something like a chemical probing experiment that only looks at whether a position is paired or unpaired cannot distinguish between these 2 structures, right? So an ideal chemical probing experiment for this and for that structure will give you exactly the same pattern. So this is one problem that we have with this probing data. The other problem is the very practical problem is that all the probing data do not measure directly whether a base is paired or unpaired, but it measures a physicochemical property like flexibility that is correlated with what we want to measure, but it's not the same thing. So this is the reason why we have to go through this whole machinery of complicated statistical predictions here. Last topic is consensus structures. So for many RNAs, we know that evolution conserves the structure, right? We know this for tRNAs. All tRNAs basically have the same cloverleaf structure as a consensus. So one quite obvious question is, can we actually use the fact that several different evolutionary related sequences have essentially the same structure? So can we use this effect of sequence covariation? And we've done that actually long time ago by modifying the RNA folding procedure in such a way that it can take, instead of a single sequence, a whole alignment as an input, and that actually drastically improves the prediction of the secondary structure. But this is something that has several shortcomings. So the idea to take the whole alignment and just calculate one consensus structure for the alignment obviously requires that you have a globally conserved structure. Otherwise, this will not work, right? So if the structures vary in parts and are only the same in some parts, you will not get a reasonable structure prediction for any particular sequence. So this assumption that we have a general consensus structure works very nicely for tRNAs, ribosomal RNAs, microRNA precursors, but it doesn't work at all, for instance, for long non-coding RNAs. They at best have local conserved structural elements. We have to assume that the structure is perfectly conserved, even in this very simple example of the U7 as nRNA from animals. You can see, well, perfect conservation looks different, right? So there is lots of pieces missing. So different animals have a different length of this stem loop structure at the end. And also not all the base pairs, you really can make an argument that they are there in all the species. So there is a similar structure at the end, but certainly not a perfectly conserved one already in this very conserved structure. So what we can do is we can again say, okay, so let's say we build an alignment. We calculate from the alignment which base pairs are very prevalent in the aligned structure. And we use then this information as a guidance similar to the guidance for the chemical probing experiments. The only difference now is from the alignments, we actually extract sequence covariation, so we extract base pairs rather than individual bases. So that actually should work a lot better. The point of this slide, so what I did here is basically to say one has to be really careful how to think about these, because often, and you see this a lot in the literature on this stuff, is that absence of evidence is mixed up with evidence of absence. So let's take a bunch of RNA alignments, just random sequences, and disturb those randomly, right? So we start with identical sequences, and then we introduce 30%, 20%, 10% of errors in each one of the sequences. So the average sequence divergence is essentially of this area, and then we throw this thing into a program like RNA-alifold and ask how many base pairs do we predict. So by construction, there should be no conserved base pair that we get, right? Because we just randomly placed mutations in our sequence, so we don't actually preserve. So there's no selection pressure to preserve a secondary structure. So any structure that we still pick up is not real, right? It's just an artifact of the fact that our sequences are too similar. So, and if we have 30% sequence variation on average, and we have 10 aligned sequences, there is actually nothing left. We've wiped out this artificial signal of consensus pairs. And the more similar the sequences are, basically the more sequences you need in the alignment to see that there is actually nothing left, that there is no signal for conservation. The point of that is that the only thing that we should believe from our base pairs, from our alignments, from the structure prediction of our alignments, is base pairs that are predicted with very high probability. And if we don't see a base pair, that doesn't mean that the individual sequence doesn't make a base pair. That just means this base is not part of a conserved base pair over the whole ensemble, right? So what we want to do now is we want to take the phylogenetic conservation of our structures. We want to take the information that's contained in the alignment, extract which base pairs are actually likely to be conserved everywhere, and then use this information on which base pairs are conserved everywhere as a way of guiding the folding of each individual sequence. So here's the protocol for that. So we start with a sequence alignment that also contains the sequence that in the structure of which we are really interested in. Then we compute the base pairing probabilities, which is to say the partition function for this alignment. We only keep the alignments with that are predicted to be with a probability of larger than 50%. And for those, we give a bonus energy that again follows this Logos law that we've seen before. If we're only interested in whether a position is paired or unpaired, so kind of mimicking the type of information that we have for from chemical probing, we just keep significantly paired positions, right? Unpaired positions we cannot get from the from sequence covariation. We only get, in this case, information about paired positions. Similar in the, as I argued, in the chemical probing case, we only get information about unpaired positions and no information on paired positions. So then we project these base pairs or these paired positions onto the sequence X that we are interested in, and then we use these bonus energies on the sequence X and calculate our structures. And this is essentially over a large set of alignments from E. coli coding RNAs. And so this is basically the performance measures that you get for RNA folding alone, which is on the order of 60–70%, which is kind of typical for our bacterial RNA structures. For large eukaryotic RNAs, probably because there's more protein stuff involved, secondary structure predictions are less accurate. But for bacteria, you actually get already, with just the thermodynamic folding, reasonably good results. And now if we include the phylogenetic information, if we include what we get from the alignments in terms of base pairings, then you get to prediction accuracies that are, and this is just done with a bunch of different methods that essentially conceptually all do the same thing in a technically slightly different way, you get results that are actually quite nice. So they're in the mid 90s of percent of the performance measure, and so that the procedure that I showed you is conceptually sort of the cleanest, I would say, and it also performs at least as well as everybody else. This is this RNA soft constant. Again, just some performance comparisons on the same data sets. So this is for various different data sets that have been looked at and with and without. So I'm just glossing over this. So the important thing, I think, is this. If we compare the effect of chemical probing now and the effect of having phylogenetic information, it's kind of interesting to see the following. So what I did in the corner here is just take the raw chemical probing data and so this is always the A, is the raw chemical probing data, nothing else, and throw them into a structure prediction algorithm. So that's the only thing now that basically I forgot about the energy rules. I just said, okay, I have these constraints of what is paired and unpaired structures, and I just take away any knowledge of the energy. And then I do structure prediction, and what I basically get is to use a technical term CREP. So this just shows that probing data alone are just not sufficient to give you reasonable structure predictions. At least it's much, much worse than just using the thermodynamic model alone. So the second column is the thermodynamic model alone, and this is simply for 3 different data sets that were selected to be fairly different in the RNAs that they choose, and they were also done with different technologies. So this is essentially a uniform feature that RNA folding does better, so thermodynamic folding does better than just using probing data. Integrating probing data and the thermodynamic model in different ways gives a moderate improvement over the for the LEDSEG, actually quite a bit of improvement. For other techniques, at least a quite reasonable improvement over thermodynamic folding. But the important point here is the last part here is when we instead use the phylogenetic information that we get from just sequence comparison. And it turns out phylogenetic information from sequence comparison actually gives you much better structure models than what you can get from chemical probing, and it doesn't matter which probing method in reality. So this is a little bit to annoy the people who are pushing chemical probing a lot, because it shows that the quality of this data is actually rather limited, and whenever you have reason to believe that you have an evolutionary conserved structure, you just get the better structure model from looking at the evolutionary data. Combining all the data, so thermodynamic probing data and phylogenetic information gives you a tiny little advantage at the end. So it does get a little bit better, but one might argue it's not really worth it. So just 2 more minutes on some stuff that's still unpublished. What we're currently working on is the question, can we actually also use data from cross-linking? So there's an increasing number of techniques for RNA cross-linking. So only look at the A part. So one protocol that we were playing around again with the Merle lab is to say, okay, so let's say we have an RNA-RNA interaction. We can cross-link this with AMT. Then we can trim this to get chemically well-defined ends, and then you can actually ligate these. You can open the chemical crosslink. Then you get this phosphate thing hanging off here, dephosphorylating these with an enzyme allows you to do to get this hybrid RNA where you basically have the position of the crosslink. So basically you have the cut here. You don't know anymore where the exact position of the crosslink is, but you can roughly get the crosslinking position. There is plenty of other protocols. I don't want to list all of them. There's at least 10 different ways of doing this crosslinking, and then either ligating these or completely on one side, or just circularizing these and amplifying it and basically sequencing these hybrid RNAs. And what you get out of that essentially is the blue interval in your sequencing product and the red interval in your sequencing product were interacting with each other. There was a helix somewhere that was cross-linked in this protocol. So this was ignore this. This was basically just a way of getting well-defined positive controls. So what we then were looking at is, well, if we have this cross-linking data, they only give us information about this interval in the RNA interacts with that interval in the RNA. So this is much less accurate than the saying what we get from the phylogenetic information, which is this base interacts with that base, right? And in a way, it's also a different type of information as from saying here is something paired or here is something unpaired. So the question that we were interested is under what conditions do cross-linking data actually give additional information on RNA folding? So do they actually improve folding predictions? And the answer is essentially yes. So this depends a little bit on how long the fragments are. So if this blue and reddish thing is very long, then it basically doesn't tell you anything, right? It just tells you there is a base pair somewhere in my RNA, which is not particularly helpful. But if you keep these two sequences of length 25 or 23 or something like this, then this becomes realistic experimentally. And you can also see, depending on kind of with moderate bonus energies that you would assign to such intervals, you actually get a significant improvement in these secondary structure prediction. Now, I have to say this is all from simulated data, because before we actually went through the pain of analyzing large-scale sequencing data sets, we first really wanted to know whether you get any signal out of it. Instead of spending a year of a PhD student's work building the pipeline to analyze the real-world data, and then find out, well, they're so crappy, you can't see anything. But this is very encouraging, and you can also see that as coverage of the crosslinks goes up, you actually get very, very good structure predictions, at least in theory. So it looks like it's actually worthwhile to use cross-linking techniques to get improvements to secondary structure predictions, right? I mean, you're up to the level, if you get 10-15% coverage of the whole RNA, you're already up to levels of what you can get from phylogenetic structure. So this is really encouraging, because what that basically tells us is that if we can get reasonably dense data, we can get to actual RNA structures, or at least reasonably close, for sequences that don't have conserved structures. So in particular, we have a chance to get at the structural ensembles of long non-coding RNAs directly, and that will basically be the next step. And with that, I'm at the end of my presentation. I would just very briefly like to acknowledge some of my collaborators. I'm not going to all the names, just the red guys. Mario Mörl is a biochemistry lab with whom we collaborate very closely on all things RNA in Leipzig for the last almost 20 years. Ivo Hofacker's lab in Vienna is the kind of our partner lab with whom we collaborate very closely on the RNA package, which for historical reasons is still called the Vienna RNA package because we started it while I was still in Vienna. And Ronnie Lawrence up here in red, he's the chief developer of the RNA package. He has a permanent position in Vienna, but is now for a 6-year stint in my lab in Leipzig. Funding comes from mostly the DFG, the German Research Foundation, and the German Federal Ministry of Sciences over the years for this work. And with that, thanks a lot for your attention, and I'm happy to take questions.
Thank you, Peter. Okay. So, some questions?
Hi, thanks for your talk, very interesting. I have a question. What is the importance for knowing the secondary structure of RNA? What is the application?
So there's several applications. So one of them, so the most simple one is to say, if I see that the secondary structure is conserved, I know there is a selection pressure on the RNA itself, right? So if you only see there is a sequence conserved, you don't know, does that pertain to the DNA level? Is that actually functional at the RNA level, or is that actually something that means conservation in a protein that's produced out of these? So this is one reason. The second point is that there is hundreds of proteins that bind RNA, and at least a third of them want specific structured elements that they bind. So knowing that an RNA is structured in a specific way can be a hint on RNA binding proteins. And the third reason is that if I look at a structured RNA, sometimes you can directly read off what it likely is, right? If you have a very stable, almost symmetric hairpin, this is likely, and you have a eukaryotic system, or this is likely a microRNA precursor. So these structures are directly indicative of their functions. Of course, if you just give me some structure, I cannot guess what the function is unless this is a structure that's has already been known. And this is not, this also goes, for instance, into viruses. There are very specific viral structures, for instance, in flaviviruses, that one can identify very nicely. They have to do with packaging, they have to do with replication, and you can also, knowing that you have RNA structures with particular functions, also makes them potentially a target, right? If you can interfere with that, with a crucial structure, say in viral replication, then that is an obvious target also for drugs, for an antisense RNA or something like this, right? So that's the sort of main point on doing this RNA structure work, apart from the entertainment value.
Hi,
Hi.
thank you for your great talk. My question is about long non-coding RNAs. I know the the prediction of the structure of long non-coding RNAs could be very difficult, more difficult than other RNAs. But there is some way, if we know the structure of long non-coding RNA, we can predict the biological function or something like that?
I would say there we don't have enough data yet to do this. What you can identify sometimes is particular binding sites for particular RNA binding proteins that may be conserved structured elements in the whole structure. So in the whole long non-coding RNA, so that is probably possible. I also don't think that computing globally the structure of a long non-coding RNA will tell you a lot about its function, because probably large parts of the long non-coding RNA can do whatever they want. Right? So sometimes I think of long non-coding RNAs more like as coat hangers that capture different proteins. So as a molecule that is essentially there to orchestrate who has to come together. That's one possible function. There's of course also these non-coding RNAs that interfere directly with the chromatin in cis, and so there's lots and lots of different things, and unfortunately we really don't have to my knowledge enough data to really look at a long non-coding RNA and say okay, so this is this and this will have that function, and unfortunately we're really not there yet, I would like to add.
Hi, Peter.
Hey.
Thanks for the talk. You said that the package can return like several structures with the same probability, basically with almost the same energy. And well, I was thinking in proteins. The proteins sometimes the final conformations depends on the speed of translation. So
Yes.
maybe that can affect also
These
RNA.
these effects of kinetic folding also do exist in RNA. So there are very nice examples of, for instance, of host killing systems. So this is basically systems in plasmids and other molecular parasites that try to ensure that they get transmitted into the offspring. So what they do, for instance, is express a poison and an antidote, and you have a time fuse on the so the antidote only works for a certain amount of time. It's trapped, so the functional structure is actually something that's trapped in a local minimum, and over time scales of half an hour or something like that refolds into an inactive ground state that then does not prevent the expression of the poison anymore. And so if you don't keep producing this intermediate, so this folding intermediate, right, this metastable state that actually keeps the poison at bay, it will kill you. So if you are an offspring that doesn't produce the antidote, you die. And so this is a way how this molecular, one of many examples where you have metastable states that are actually the functional thing, right? And the ground state is actually not functional. But in general, you also have this effect that RNAs can be trapped essentially forever in a kinetically determined state, and very slowly or not at all for practical purposes reach the ground state. But this is something that with this technology we cannot look at. There is other ways of doing this where you have to basically look at all the metastable states, kind of enumerate them like I showed on this slide, and ask what is the energy barrier from one to the other. So we've done some work in that direction. This is for smallish molecules. This is actually possible to find out whether there is kinetic trap states that you have to take into account to understand the molecule.
Hi, great talk. I was wondering about the salt concentrations that you mentioned before. These concentrations are in the range of the cellular levels, or
Yeah, so
but my question is about if you have different types of cells, how these concentration levels varies or have different concentrations in different cells. So maybe you can have different regulations by the by the fold of the RNAs?
That is possible. So I don't know enough about the physiological data on how much, say, salt concentrations vary between different human cells. But if you think about, you're interested in RNA structures, say in a group of bacteria that lives in very different environments, from say soy bacteria to bacteria that pelagic bacteria to hypersaline species that say live in the Dead Sea or in Quattro Ciénegas or somewhere, then you easily can have probably 2 orders of magnitude in difference in the salt concentration. So these changes actually do play a role in practice. What I didn't belabor, because that was built in for a longer time, is that RNA folding energies and RNA structures are also dependent on temperature, which is also built into the software, and it makes a difference on whether you have cryophilic bacteria that like basically to live on the frozen on the surface of icicles or between ice crystals in the Arctic, or whether you have something that basically lives in an almost cooking geyser. So 100 degrees also make a big difference, and you will also see this actually in the RNA, because cryophilic bacteria have much more AU on average in their genome, in particular in the genes that code for RNA, while hyperthermophiles are much more GC rich, for instance.
Yeah, but I'm asking about maybe in other organisms more complex with different kind of tissues.
I simply don't know I have no idea what the actual variation of salt concentrations between different human cells is, or how much that can change, not only between different cell types, but also depending on how much salt you eat or whatever. I simply don't know.
Okay, yeah, thanks.
But it's an interesting question, obviously, to look at this.
Okay, thank you very much, Peter, again for this presentation and the questions. So now, Peter is teaching today the RNA structure, so we hope to have fun with him. But I think because the talk was a little longer than other days, we go first for coffee break to relax.